It's true. Thank you. Show like this. Another. Oh, okay. No, it's fine there. Okay, morning everyone. Thank you, Tadiwa, for the introduction. So today I'll be talking about the growth and diversity. I think it's fine.
It's fine.
You go back. No. It's fine. It's fine. Oh, okay.
Yeah, better.
Yeah, sure. So today I'll be talking about the growth and diversity of NRF-SAIAB collections. Can you come in? Not with the slideshow there. Right? Slideshow. Sorry about that. Or if I click here,
Sharon.
Yes, you have put the slideshow here. Yeah.
I haven't seen.
I can't. Don't slideshow of things. Slideshow of things. It's not true. Because it's not true. If you click here, it doesn't.
Okay, so whilst we are trying to figure out the technical difficulties that we're having, I thought maybe I just share a bit about the International Day for Biological Diversity. So essentially, this day was established by the UN on the establishment of the international.
This presentation.
So it was made to commemorate the formation of the Convention for Biological Diversity. So it was established in 1993 on the 29th of December. But instead of celebrating on the 29th of December because of all the other holidays that are in December, they decided to move it onto the 22nd of May. That's why we celebrate on the 22nd of May. But it's actually a commemoration of the formation of the Convention for Biodiversity, which was on the 29th of December 1993. And this year's theme is be part of the plan, which is essentially a call to action for all of us to be part of the reversal of the loss of biodiversity within, because of climate change and anthropogenic effects. So essentially, we are being called to action. And as part of being part of the plan, our speakers are going to talk about some of the work that we have been doing, particularly with the use of the collections, how storing materials in the collections and how that material can be used in order to reverse that, as well as work that we are doing as part of the freshwater group. So I think now that the technical difficulties have been overcome, I can hand over back to Nonkoliso. So once again, I'm introducing Nonkoliso Mgibantaka, who is a collections officer, and she will talk about the collections. Thank you.
Thank you very much. So today I'll be talking about the growth and diversity of NRF-SAIAB collections. So I thought it would be great to talk about this day before I go to my presentation. So this year's theme says, be part of the plan. Be part of the plan to reverse the loss of diversity, to protect our environment, to restore whatever we have, to reduce, to reuse and recycle any waste material, to share the benefits of the environment. That is, we use resources in a way that our children and our grandchildren can use the same resources. And we are doing this collaboratively for the nature. NRF-SAIAB's vision and mission is to improve the quality of life of South Africans by advancing aquatic research. We strive for excellence in research for the sustainability of the environment. NRF is responsible for the conservation and protection of our natural environment, that is marine and freshwater environments. Why do we need this collection? The aim of National Collection Facility is to provide access to the specimens and to the data. The activities that we do in CMC are acquisition of new material, sorting and cataloging of specimens, care of specimens, making sure that they are well preserved and protected. We support collection users through loans and facilities. Why is it important for us to have this collection? It contributes to education, to IUCN RED data assessment, to development tools such as environmental impact assessment, water use licenses, biosecurity, management of biodiversity, and that of invasive species. The activities that we do in CMC are governed by rules and regulations. We have policies and SOPs in place. In order for you to collect or bring specimens to SAIAB, you have to have these documents, the permits, the ethics clearance. If you are donating any material, you need to fill in a donation form. This is the CMC staff. What I want to emphasize here is teamwork. We are working together as a team for a common goal, which is to serve you guys with the specimens and the information. So that teamwork makes it easy for us to work with you, that is the universities and students, the scientists and museums, both South Africans and non-South Africans, the government and NGOs, and public in general. The history of our collection of SAIAB, it started as J.L.B. Smith Institute of Ecology, and they focused on fish only. And then when it changed to NRF-SAIAB, we included all aquatic animals, not only fish. This is the map that shows all our records, all our specimens that we have were collected from those areas. The history of our collection, it started, we used to use the library as our collection building. Then because of the growth of our collection, we had to move to the new building, which is the one that we are using, which looks like that picture, all right? We have different types of collection. We have national fish collection, which includes both the wet collection and dry collection. We have amphibian collection, invertebrates, our biobank, that is tissue collection. Sometimes we don't have to bring specimens here. We just take images and tissues and release the specimens back to their natural environment. We have millions of specimens here at SAIAB, which are made up of 522 families, 126,467 number of lots, that is your jars and tanks. And then we have approximately 45,000 tissues. I will explain later why we have few tissues compared to voucher specimens. Our fish collection is dominated by two families, that is your Cyclidae and your Cyprinidae. Our collection is arranged alphabetically according to family, genus, species, catalog number, and lastly, your jar sizes, your jars. It's arranged alphabetical from family Acarithuridae to Zoachidae. The reason why we have few tissues compared to our voucher specimen, biobank was introduced later. So I think Roger is the first person to start taking tissues from the fish, and he used to keep those tissues in his office or whatever. And then later on, they bought that freezer, which is the normal freezer that we use at home. But now we have 8 freezers, 2 chest freezers to temporarily store the tissues, and then 6 upright freezers. Because of the changes and the growth, we're no longer using this register book. We are moving with moving times. Yeah. So we are using Specify now as our database. And because of the change in the technology and stuff, we have X-rays now. We have photo, we have a studio, Zinzi and SPA are busy there taking pictures. So there is a slight shift from physical specimens to the visual piece. The highlights of our collection, we have our oldest specimen, Sandelia capensis. This specimen is one of the specimens that were donated by Albany Museum. It was collected in 1875. The second oldest specimen is this one. It's from United States of America. It was collected by David. I'm not sure if this one is the third oldest specimen. I chose it because it is the first specimen to be recorded to our database. As you can see, its SAIAB number is 1. It was collected in 1967 by J.L.B. Smith. And then we have our iconic specimens. All our visitors always ask about these 2 specimens, the silicans and the albino great white shark. What is special about this great white shark is that it is an albino. So it's not easy for it to hide from predators. So this one is special because it managed to grow to this size. And one other thing that is special about our collection is that we have IUCN red listed specimens. Those that are vulnerable to extinction, they are threatened, they are endangered. So even if they go extinct in future, we'll still have them in our collection. I'm not sure if we do have any specimens that are extinct in the world. I'm not sure, that would be great. And then improvements and achievements. We are so fortunate that we are the one of the institutes that joined NSF, which is National Science Collections Facility. So most of us have managed to do this course, which is collections management course, and we had opportunities to attend forums and conferences. Sesh attended a conference last month, which is one of our achievements, and then improvements in terms of infrastructure. You know, safety comes first. So we just updated HVAC. We constructed band wall. We have a building monitoring system, the backup systems, recyclers. Remember, I mentioned in that slide that we reuse, we recycle, so we don't have to buy ethanol every time we need ethanol. We just recycle our ethanol. And then I praise my collection, but if there's no one using our collection, it's useless. So we do have collection users. These are the loans, voucher loans for this year and last year. So as you can see, I highlighted external loans. That is people from outside. As you can see, there's specimens that were sent to Australia, Belgium, Russia, and other countries. And these ones are tissue loans, as you can see. So we do have collection users. And then your PhD is not your PhD if you don't share your information with South Africans or Africans. So you remember SAIAB's vision and mission, it says we have to improve the quality of life of South Africans. So if you don't share your information, that means you are not making any improvements to South Africans. So these are publications. The first row is South African, that is Albert Tadiwa. I mean, they used our connections. They were, it's Tadiwa. You published this last month, Tadiwa, but it's this year's publication. And then there's Albert, there's Ionela, and then we have Pedro, Stoffa. The last one is Abed. Yes. But we have challenges. We have orphaned specimens. We have few taxonomies. We have backlogs. We have this big collection of cephalopods, and we don't have a specialist to identify that. So we always ask Lipinski. He's very, very old. So if something happens, we want anyone to take over. So I think I challenge you students to think of learning how to identify cephalopods. Not only students, even as collection, you can do that. You'll never know. You see things are changing. And then we have few taxonomies and backlogs. Sometimes what happens is that the researchers go to the field, they bring specimens, and then they go again, they don't sort their specimens. I think the solution would be for them to go to the field, you sort your specimen, and then you go to your next field trip to avoid those backlogs. Where to from now? That is the future. We will continue building on our collection, and also the biobank. They are focusing on collecting more frogs and invertebrates, including the marine sponges. It has been discovered that there is this chemical which is produced by sponges, and that chemical is good for the treatment of diseases. So they are. And then we are planning to buy a CT scanner. So as I've mentioned, there are a lot of changes. There is a shift of focus from physical to visual. So I'm challenging all of you guys to think of your careers, to upskill yourselves, to capacitate yourself, so that even if in future we no longer need voucher specimens, at least you can still fit in. So we need more taxonomists. Yes. So thank you very much for listening.
Thank you very much, Nonkoliso, for that very interesting talk. And I'm sure there are quite a number of questions that are burning. But for now, I think we'll move on to the second speaker. Then we'll just have one question and answer session after that. So the next speaker is, like I said, Dr. Scheepers, and you'll be talking about today. I don't know what he has for us, but I'm sure it's going to be interesting.
Okay, morning everyone. Thanks for coming to listen to our talks. Mine today is a pretty broad introduction to what biodiversity is and why it's important, and why it's important to look after our biodiversity on the planet. So what is biodiversity? It encompasses all life on Earth, from your microbes to your fungi to your animals, everything that's ever lived and evolved on this planet, is what we call our biological diversity, or biodiversity for short. And we kind of talk about it in three different components. Talk about genetic diversity, species diversity, and then ecosystem diversity. Now, genetic diversity just means, you know, obviously the higher genetic diversity in certain populations than in others, and that's got some adaptive benefits in larger populations with or species with higher genetic diversity. They tend to be able to be more resilient to external changes, changes in the environment, and so forth. Low genetic populations or species with low genetic diversity, because they do not have many variations in their genome to adapt to climate change or habitat disturbances, they are more prone to be in trouble when there are changes. Species diversity, just again, that's all life on Earth. We've probably described about 1.5 million species so far, but they reckon that there's at least another 10 million that needs to be described. And what this kind of also means is that there are a lot of organisms out there that we have got no idea what they are at the moment, and what their function is in the ecosystems that they live in. And by kind of looking after even the things that we don't know about yet, we might be able to, you know, find some kind of a benefit in the future. And on a community level, you can describe species diversity there on the right. Higher diversity, it's just got more species than community 2. And then ecosystem diversity just refers to kind of different ecosystems or habitats that all these organisms live in. And ecosystem diversity is just as important as your species diversity and your genetic diversity, because organisms, they all need to live somewhere. But what has been happening for biodiversity in the last, this is now the last 50 years or so, we've seen a significant decline across the world, kind of differences between areas, but I think the average in the end is around 69% decline of abundances and species diversity. And this is only from 1970, from when they kind of started keeping this record. And so a lot of this was lost. A lot of our abundances and species diversity would have been lost in decades and centuries prior to this. So you can see that there is a significant decline throughout the world. And what drives this decline? Probably humans in as the starting off point, population growth, increased consumption, reduced resource efficiency. So we are driving habitat loss. We are instigating invasive species that get into new areas. overexploitation, think of, deforestation and overfishing. Pollution is a major one. And then climate change and its associated global warming. All of these factors lead to biodiversity loss at the genetic level, organismal level, and at the ecosystem level. But then, why is biodiversity so important to us? And the simple fact is that it sustains human livelihoods and sustains life on this planet. There's a number of larger themes where biodiversity is important. One is food security, there's solutions to climate change, there's for human health, and then there's cultural and economic benefits to higher biodiversity. And I'll just touch on a couple of these. And so all our food comes from the natural environment, whether it's farmed or wild, caught or collected. And in terms of even agricultural crops, we've seen a decline in the number of crops that's been grown around the world. In Southeast Asia, there used to be about 50 plus varieties of rice that they used to cultivate, and in some areas that's down to 2. And then you can start seeing if some kind of a pathogen or something were to get a hold of one of those varieties, it might disrupt a large portion of the food supply in that part of the world. Many rural communities rely on fishing and wild-caught foods, and this leads to higher biodiversity. It's more things that you can eat and more nutrition. I mean, our nutritional problem is probably by eating too few kinds of foods, over reliance on certain crops. There's economic values to increase biodiversity for raw materials, wood, fibers, clothes. Many of these things come from the natural environment. Some medicinal plants, and with that also plants of other economic values. Think of essential oils, rooibos tea, aloe vera. It's got a health benefit according to some, and economic benefit. And in terms of the economy as well, this is kind of an infographic from Africa. And you can see that the biodiversity-driven sectors, fisheries, recreation, carbon sequestration, water purification, food production, everything that's got a very large economic value. And like at the bottom there, you can see in Southern Africa, they estimate that the recreation value of biodiversity-related ecotourism is around $11,000 per square kilometer per year. And this directly leads to job creation and economic security. Then other important factors of better increased biodiversity is nutrient cycling. In a higher biodiversity system, nutrient cycling is it's increased. Water purification, wetlands, the protection of catchment areas where high biodiversity basically purifies our water for us. And this has knock-on effects for water security, decreased economic costs for water treatment plants downstream. And without these networks, water security would go down. You'd get an increased nutrient runoff downstream. And the importance to human health. So our COVID pandemic was also linked in a sense to biodiversity loss and ecosystem health. Habitat degradation, the illegal wildlife trade, means that there's an encroachment of humans on the habitat of a lot of species that harbor zoonotic diseases, and our closer association with them increases the likelihood that there might be a spillover and a jump from animal vectors to humans. And as our deforestation especially goes on, there is increased likelihood down the line of maybe more severe pandemics in the future. And then we derive quite a few products for medical purposes from the world around us. We've touched upon sponges just now. They've got compounds that are used in cancer treatments, chemotherapy. There's snails that have compounds that's been used for pain relief and antimicrobial properties derived from the natural environment. On the right there, you can see fish skin. That's tilapia skin that's been used to treat burn victims, and apparently it works pretty well. So from SAIAB's perspective, in our biodiversity research that we do, especially in the freshwater group, we do foundational work, classification, taxonomy. We do species assessments, those red list assessments, which then increases our knowledge on the biodiversity of the systems we're working on. From there, we then build into policy and talking to government about conservation and so on. And then what we try to influence ultimately is then human well-being. And to tap it off, a little quote from the legendary E.O. Wilson, saying that biodiversity is the totality of all inherited variation in life forms of Earth, of which we are only one species. We study and save it to our great benefit, and we ignore and degrade it to our own peril. Thank you.
Thank you, Martinus, for that talk. So now I think we'll open up to the floor and to online for any questions for both Martinus's talk and Nonkoliso's talk as well. So yeah. So I'll start with the floor. Are there any questions? Yes, Matt.
For Nonkoliso, are there any drives in South Africa to increase the taxonomic skills in the country?
Are there any what?
Any efforts in the country to grow our taxonomists, right? To train new taxonomists, increase the pool of taxonomists? To identify that as a problem, but
I don't have an idea. I don't know why we have new taxonomies. I'm not sure if most of the people don't even know what is there, or they're only exposed when they reach this level, when they're here, I don't know. Or maybe I'm not sure if government is working in advertising taxonomy and stuff.
is it something the NSCF could drive, create more awareness, perhaps fund from students?
I'm not sure if something is.
Yeah,
doing a great job because most of the grants that they are giving out to people who want to do taxonomy. And also most of the people that are coming to SAIAB to work on the collection, most of them are funded by the event. Like Nonkoliso is mentioning now, Martinus Institute donated his entire collection. But then again, we want to sit down and think this, because this guy has been working for how long in his entire career. How many people does he, produce? Because I think you need to produce people, like look at Albert, for example. So when he retires, we can happily say at least there's about maybe 10 people who would be able to do the kind of studies that he has done. I think the marine guys, someone who can advance, taxonomy in marine. So we need, we definitely need that. And for other communities that we're also talking about that, so collections that do not have curators, they will never grow. They will sit there idle. They need to have someone who is actively on the ground working on them. Like our cephalopods, I don't think they will grow. In our flow collection, Roger is still there collecting, but who is, we need to have curators who will push taxonomy and collection using of that collection. Yeah, so there's a lot of, look at SAIAB, we're growing our own team of taxonomy.
Yeah. We do well, but
Yeah.
is there a greater national
interest? Yes. I think someone is pushing.
Okay, thank you very much.
We have one.
I think most of them are plant taxonomies. I think most of them are plant taxonomies. The problem is fish taxonomies.
Do we have them in South Africa except SAIAB?
No.
Just asking.
Except saying. You don't have plant. Plant taxonomies. No fish. Or.
We have them.
Not outside SAIAB. Outside SAIAB. No.
Okay.
No one. Outside SAIAB.
in the marine waters about. Janelle has done quite a bit of work. Probably the only young.
Yeah, I'm raising this precisely because as an institute, this is what the impact. You see, when you go to strategic level, you want to say that what am I producing? What impact does it have in the global or local or provincial structure? And then, and if that has been identified that there is a gap in opportunity, then how then do we attract funding so that we can then be able to produce more? Not necessarily that will be housed inside, but that will work across the globe and everything. So when you have this discussion, it's very important. Also, I think we've done in the ROV on the risk register that if what happens if Ryan is not around, what contingencies do we have in place in a space of a biobank? That is where actually you need to discuss as peers that have capability to build your capacity around that and becomes an opportunity. And those opportunities should be documented so that when the funding was requested, and then it should be reflected to say that there's an opportunity for this and this needs to be done at the global scale. That was my suggestion. So let's have a nice discussion by and then my next question before I leave. I see in Nonkoliso's discussion, you're talking about NGOs. Do we have NGOs? Who are those NGOs? And how do we pursue in pursuit of governance relations? So that when we get the funding, as I said, 90% of the funding comes from DSI. How then we influence locally, provincially, and nationally, so that the interest and the investment should come. And SAIAB should be different. I think that's what Engels would always teach you that. B, not produce science. Science must have an impact. And who's your funder? How do we attract that funding? So that when we discuss those things strategically, you would then be able to say that who are our NGOs? Do we have an NGO locally? Yeah, in Grahamstown. Who are we working with? Do we go to the school? Do we go to, you know what I'm saying? And then how do we then have an impact on a provincial scale? Who are the resources that we have? For me, you know, Nonkoliso, you can just allude except NCS and NSF. Who's your other NGO? How do we self-sustain without getting money from government? If that very well to get NSF, who else might want to fund you guys? BBSA, what relations do we have? So those are the discussions that I would want us to, at a very strategic level, when you discuss this, it was right to say that that's how we attract the funding and keep ourselves sustainable. That's my comment.
Thank you very much, Tabo. It's actually a very interesting discussion and a very important discussion that I think we ought to continue having even beyond this platform. There's a question on the Q&A online, then open it up again for the floor. So this is from Punza. How many species have gone extinct in South Africa? How many species are endangered in biodiversity hotspots in South Africa? I don't know.
I would probably have to look that up. Off the top of my head, it would be quite difficult to say. I mean, fish-wise, there's a good, I think, 10 at least that's endangered in freshwater fishes. Extinct, it's difficult to classify something as extinct.
There's three extinct in Africa.
In Africa,
According to the IC
Mm.
extinction classification.
I mean, then there's a few of the other mammal species. If you think of the quagga is gone, and I think the Cape lion is gone.
The sawfish as well, South
Sawfish.
Africa, they've disappeared from South Africa. Easy fish down.
Even that, I'm not sure. Guilty.
And there's Labiobarbus Worthingtoni, I think, is also.
I think she can rate the IUC.
Yeah, go check on the OCN.
Yeah, so just to add to that, like there are a couple of that have gone extinct, and there are other species that, beyond their initial descriptions, have not been recorded since. So essentially, that is work that still needs to be found out, whether some of these species are still present or have been forgotten. Yeah. Okay. Any more questions from the floor? I think that's since there are no further questions, I think we can conclude our talk. Don't forget there are posters there as well, so please do, before you leave, just have a look at the posters. And I think the people who made the posters are within the group as well, so they'll be there to answer any questions you may have. Thank you very much for coming to our SAIAB seminar, and thank you very much for participating.